So, hello everyone, my name is Luisa Vargas Cassol, and today I'll talk a little about my work, discrete modeling of epithelial-mesenchymal transition, the whole of no-coding RNAs. Actually, I will focus on some initial concepts. So, let's go. Proteins. What is a protein? Proteins are large biomolecules that consist in one or more long chains of amino acids that make possible the most biological process in a cell. And it includes the DNA replication, cell growth, proliferation, and apoptosis. Apoptosis is a programmed cell death process. And the proteins, they can interact with each other, and it can be a transient interaction to regulate some specific effects in a short time. And it can be also permanent interactions to form stable proteins complex. Here I put a representation of the structure of one protein. This protein is myoglobin. I just take from Google. So I already talked about it, but again, EMT, epithelial-mesenchymal transition, it's a biological process where epithelial cells are converted into mesenchymal ones. So during the epithelial cell, they undergo this mechanism, losing their adhesion properties and gain mobility. So during this process, the cell can be temporarily stable in 3 different phenotypes: the epithelial one, the mesenchymal one, and one intermediate phenotype called hybrid. And in this hybrid phenotype, the cell have both the epithelial and the mesenchymal features. So a gene regulatory network is a collection of small molecules that connect with each other or with other small molecules in the cell to control the gene expression. So in other words, the gene regulatory network is responsible for controlling of the biological development of the cell. So here an example of a regulatory network is a regulatory network for G1S checkpoint pathway. So we have the nodes that are the molecules on this network, and we have the interactions that the molecules are having. So we have the interactions can be of inhibition and can be also positive interaction. I forgot the activatory interactions. So to simulate these networks, we have ginseng. Ginseng is a software of modeling and simulating of regulatory networks. With ginseng, we can analyze the dynamic of the system. So we have some tools on ginseng, and one that I use a lot is stable states. So we can analyze the stable states of the system. Here I put a example of this network here, that we see the stable states of all the molecules. The S is the input, that is DNA damage. Another tool that I use a lot is perturbations. So with perturbations, we can make perturbations of the system. So we can cut the nodes or we can the sign of the nodes or the sign of the interactions, and we can also increase these nodes or increase the sign of these interactions. Thank you. So here I put my regulatory network that I'm working. Is based on a network of or it's based on another work of our group. And here I am studying the influence of these long RNA, long non-coding RNA ATP, and also the influence of ECM. ECM is the extracellular matrix. And what is the effect of these two in the EMT? And I'm looking for the hybrid states because in cancer context, the hybrid states in EMT are more aggressive than the other states, the epithelial and the mesenchymal.